But I'm happy to introduce our colleagues from UC San Diego, Audrey Tremblay and Tina Chambers et al. I'm sure there's going to be many people speaking. So Audrey, take it away.
Thank you, Prince. And good morning or good afternoon, depending on your time zone. I'm Audrey Tremblay, the contact PI for our imprint grant here at UC San Diego. And I have the pleasure to introduce our team. So you can go to the next slide, please. So in terms of structure, I'm going to show this graphic, and you'll see pieces of this graphic throughout each project and core talk. And so just to kind of give you a lay of the land of how we are looking at things here at UC San Diego, our work here really focuses on that first picture there with the mother, infant, human milk circular diagram. That is at the core. It's held by a clinical project that Tina Chambers oversees. And then we have there in the middle our data science project that Shirley Sonoda and Peter Dorstein oversee, that has crosstalk between the human side, and then on the right side there, our basic science project led by Victor Nazay and George Liu, who you'll hear from today. Two cores that we have supporting this work is our milk analytics core led by Lars Bodie. He's not with us today for a very good reason. He had a new addition to his family this weekend, so I will be pinch hitting for him. And I'm not a replacement, but I'm happy to pass on questions that relate to the milk analytics core that we may not be able to answer. And then finally, led by Jeremiah Mompers and Brookie Best, we've got our pharmacometrics and analytic chemistry core. Go on to the next slide, please. Administratively, the way that we are set up here is that myself and Tina, as part of the executive committee, interact closely with the KRCC and also oversee our support pool project, which you'll hear more from our support pool project tomorrow from Shirley Sonoda, as well as Sydney Thomas. Sydney actually is a T32 NICHD funded fellow for the T32 that I actually oversee for UCSD along with Victor Nazay. And then you see there are three projects as well as the two cores that I already addressed. So this is kind of the setup of our team and how we interact with the KRCC. Next slide, please. So I'll just very briefly go through the admin core here. I mean, really we're set up, the administrative core really had set up here to make sure that the communication flows well, that we've got critical evaluations that are happening. We interact with our CTRI's evaluation team. Of course, the difficult task of financial tracking. But probably the more fun part of the admin core, and I would say the more relevant scientific part is that we do administer the support pool project. Again, you'll hear from Shirley and Sydney on that tomorrow. We focus a lot on outreach and dissemination, both within the patient side of things, as well as scientifically, making sure that our work with imprint is getting out and about. And then really focusing on the key training experience that we're all really invested in. Everyone on this call is invested in terms of creating the next generation of folks that work within this field. So with that, next slide, please. So I'd like to hand it over to Tina Chambers, who oversees our clinical project, so she can run through that. Tina?
Thanks, Audrey. So as you saw in the initial schematic, there's a clinical project fitting with the theme of antibiotics use. And so this clinical project has three aims. The first is to determine the pharmacokinetics of 4 commonly used antibiotics in human milk, to look at the impact of these antibiotics on milk composition and human milk oligosaccharides, as well as microbiome profile of the mother and infant, and then to explore the relationship between those antibiotic use during breastfeeding and growth and development in infants breastfed by mothers treated with antibiotics. Next slide, please. So we're using the existing infrastructure of the UC San Diego Human Milk Research Biorepository, which is a data resource that was established in 2014 at UC San Diego, but is a US and Canada-wide biorepository that enrolls individuals who are breastfeeding, who provide a wide variety of clinical data, provide milk samples, and then allow us to follow them and their children up to about 3 years of age for growth, neurodevelopment, et cetera. And so we're using this existing research biorepository to enroll at baseline about 480 women. We're focusing this on San Diego County, primarily for budgetary reasons, because shipping costs from across the country add a significant cost to the project. So we're focusing this locally, selecting women who are breastfeeding infants who are less than 4 months of age. And then in order to identify those who subsequently are incidentally exposed to antibiotics or take an antibiotic, we have set up a process of weekly follow-up once they've enrolled, provided the baseline data and samples. We contact them weekly via automated text to query about their breastfeeding status. Are they continuing to breastfeed, and have they been prescribed a new antibiotic? So from that group, we anticipate identifying at least 10% of women who are prescribed a new antibiotic, and enrolling women 40 or more who have exposure to one of the target antibiotics, 40 who are age matched in terms of their infants who come from that initial 480, but who have not been prescribed an antibiotic, and then an additional smaller sample of women who are not breastfeeding currently, but are prescribed an antibiotic. And from those who are breastfeeding, we're collecting 3 additional sets of samples, and those who are not breastfeeding samples as well. Next slide. So exclusion criteria include, has the mother been prescribed an antibiotic since delivery and prior to the time of enrollment? So prior to the baseline sample collection. She has to be breastfeeding a singleton. She can't, for the antibiotic treatment, it can't be for mastitis, and a few other minor ones. Each participant is provided at baseline, and then subsequently, if they participate in the sub-study, a kit and instructions to self-collect a milk sample of 1 to 2 ounces. They're also be asked to collect with the kit that's provided infant and maternal stool samples, infant and maternal skin swab samples, and environmental swab samples that include kitchen sink handle, infant bedding, maternal pillow, and maternal cell phone. And participants receive compensation for their time in providing the samples. And all of this, these samples will be used in collaboration with the data science core, with the milk analytics core, and the pharmacokinetics core. Next slide. So this is the schema that shows the sampling timeline. So those who are breastfeeding provide a baseline sample, fecal skin, breast milk and environment, and the fecal and skin samples from the mother and the infant, who subsequently start an antibiotic, then provide additional fecal skin, breast milk, and environment samples over the course of the antibiotic treatment. And it's designed to align with the specific antibiotic and that individual course of treatment, working with Dr. Momper to set up that timeline. And then, as I said, for those who consent to the sub-study, they're immediately matched one-on-one to someone at baseline who has not started an antibiotic, and they're consented to provide that same set of samples over the course of their paired antibiotic treated mom. And then the ones who are not breastfeeding, who are prescribed an antibiotic, provide, of course, all of the samples except for the milk samples. Next slide. So for the follow-up, in addition to the milk samples, the mother releases medical records for the child up to 1 year of age, and they're abstracted for growth and for evidence of serious and non-serious infections in those children. Consistent with AIM-3. And in addition, when the child is about 12 months of age, the mother is asked to complete the Ages and Stages developmental screening questionnaire that is sort of a standard tool used in that age range. Next slide. The sample processing is obviously in collaboration with the cores. So Dr. Momper and Dr. Best are looking at determining PK for the specific antibiotics that are reported. Dr. Bode is looking at human milk oligosaccharides in samples from those who are antibiotic treated and their matched untreated pairs. And then Dr. Sunoda and Dr. Dorstein are looking at the metabolome from milk as well as the environmental samples. And then the microbiome will be looked at the stool samples as well. Next slide. So where we're recruiting, the typical resources where we recruit for the human milk biorepository in general include social media, clinical sources. Since we're focused on San Diego, we put staff in our general pediatrics office where they're recruiting people who are coming in for well child visits, who have an infant under 4 months of age, through referrals from the newborn nurseries and through our donor milk bank. And then direct to consumer through social media and direct to consumer ads, which has been a highly productive way for us to recruit individuals into studies. And then we have an ongoing what we call mother to baby pregnancy studies that are nationwide, but recruiting people who are enrolled in a pregnancy study who go on to breastfeed, who are residing in San Diego County. So next slide. So the text, this is the first time we tried to do this, setting up this automated text to query to identify a new exposure for people who are enrolled in the baseline study. So the text simply asks for, are they still breastfeeding and have they been prescribed a new antibiotic? And if they respond in a manner that meets the criteria, they're still breastfeeding and they have been prescribed an antibiotic, an alert email is sent to the study coordinator. And then right away, within 24 hours, the staff can contact that participant for enrollment into the antibiotic sub-study so we don't miss the opportunity to collect that sample right at the time that she's prescribed the drug. And then the text answers are integrated into the study database in real time so we know where we stand. So next slide. We had somewhat of a delay in getting started with IRB approval, but now that we're out in the field for the last couple of months, we've enrolled 36 moms at baseline, and consistent with what our estimated that we would have about 10% who would qualify for the sub-study who have been prescribed an antibiotic. So far, 3 have met that criteria. So we'll move forward over the next year or so until we meet our baseline sample of 480. And we're also, we set sort of an estimated top 4 antibiotics that we thought would be the most common ones that will be used, but we're really kind of agnostic to what that might turn out to be. So although we said it would be amoxicillin, Augmentin, nitrofurantoin, and one more, it may turn out that we're getting more cephalosporin type antibiotics. So we'll just go with what it is that we are able to accumulate sufficient numbers of. So I think that's the last one. And I'll turn it back to you, Audrey.
Thank you, Tina.
Hey, I'm Victor Nizet here. I hope you can hear me. I'm actually in Washington, D.C., just wrapped up the ASM meeting, and I got kicked out of the time limit for late room discharge into the hallway. So this basic science project is between me and George Liu, who's also a physician scientist on this call. George is the chief of pediatric infectious disease in our department, and I'm the vice chair for basic research and a chief of a division we call post-microbe systems and therapeutics, where the pediatric pharmacology participation all the way back to the PPRU and the RPDP has been, and Audrey, Peter Dorstein, and Brookie are part of that project. So our work here is focused on perinatal antibiotic exposure, and in particular, antibiotics are administered to protect infants, humans, from the leading cause of severe invasive bacterial infections in infants, which is group B streptococcus, which can cause bacteremia and meningitis. Since the early 90s, there has been universal screening of women during pregnancy for GBS colonization to guide, when positive, the administration of antibiotics during labor. And if you get more than 4 hours worth of antibiotic coverage, your risk of early onset group B streptococcal infection drops almost tenfold. So that certainly had a very big human health impact, but we also believe there is increasing recognition of the downsides of large-scale antibiotic exposure and some discussion as to the risk benefit ratio. The other thing that we know is that there are clear benefits to breastfeeding, and science suggesting that the diverse oligosaccharide component of the breast milk is a major contributor to not only infant health in terms of growth and nutritional status, but the developing immune system and the richness of the microbiome, which themselves are very well intercalated. Next slide. So when intrapartum antibiotics are administered, and approximately 10 to 15% of women in our country carry group B streptococcus, and like most bacterial infections, especially those of childhood, the developing world is disproportionately affected, and universal screening programs are certainly not the norm. You have a significant effect on the diversity of the microbiome, and beneficial microbes like the Bacteroidetes and Actinobacteria, including Bifidobacteria, are decreased, and there's an increase in Proteobacteria, which are associated with more adverse health outcomes. And this dysbiosis and exposure perinatally to antibiotics, either in the NICU or even through GBS prophylaxis, has been linked epidemiologically to increased risks of obesity, of asthma, and even of intimal thickening in the aorta, very, very early precursor of potential cardiovascular risk long term. Next slide. So in some work that we did together with Peter Dorstein and with Rob Knight in our division in the lead up to this proposal, we have set up a system where we can basically measure the molecular content through unbiased shotgun metabolomics throughout the body of an animal. Basically, we can rasterize an animal into 96 parts to be analyzed in a multi-plate format, and Peter can describe in more detail the fine aspects of the mass spectrometric analysis, but we can pair that with 16S ribosomal RNA analysis. And we first did that to just compare notobiotic mice to normal mice, and found differences in molecules throughout the body, including the discovery of dozens of never before recognized bile acids, which were modified by the flora. And then in a follow-up to that data set, we administered single doses of antibiotics, ampicillin, which will be the key study here, and vancomycin, and found changes not only in the microbiome throughout the gut, but in the molecular composition, in the metabolic analysis, predominantly in the gut, but also in distant organs like the adrenal glands and the kidneys and the uterus. So you can imagine that any biological process that's going on, whether it's a homeostatic process or a disease process like inflammation or a host pathogen interaction and trying to clear an infection, is going to transpire differently when the molecular environment is altering. And we know that we don't just give a single dose of antibiotics. Next slide. Another thing that is very interesting related to the milk oligosaccharides is that some of them show direct bacteriostatic activity against newborn pathogens. So in addition to the nutritional benefits to the host and their ability to serve as prebiotics for some of the normal flora, these particular oligosaccharides, which are abundant in human breast milk, directly inhibit the growth of group B streptococcal strains. And this was actually subsequently confirmed in a paper from the outstanding researchers there at Vanderbilt. Next slide. Breast milk may also have other things that benefit the host pathogen interaction, as we showed that HMOs block the pathological invasion of E. coli and the cytotoxicity E. coli had, for this example, on bladder epithelial cells, because there have been clinical data linking breast milk consumption to lower risk of urinary tract infection in the neonate. Next slide. So our proposal involves this type of experimental design. We're going to model 3 types of antibiotic exposure, and we are doing either timed pregnancies, in which we then administer ampicillin to the mother kind of during the last day or two, which resembles the type of parenteral treatment that a woman would be getting in pregnancy if they were GBS positive. Second, and this links in with the science of Project One just described by Tina Chambers, is ampicillin via breast milk, meaning that the mother is going to be treated after delivery and the infant would be exposed through the breast milk. This might be modeling treatment of a perinatal complication in the mother. And finally, modeling what happens when an infant is born and then find themselves with some symptoms around delivery. Often empiric antibiotics are administered to the infant for fear of bacterial sepsis, but there is data that for every one infant with true bacterial infection that is given antibiotics perinatally, there are 10 additional babies with no bacterial infection who receive such antibiotics. And often these empiric regimens are extended 2, 4, 7 days. And that's a lot of antibiotic exposure with ampicillin, a very broad spectrum antibiotic. You might imagine that it's very hard for the normal population of the gut flora to occur in that kind of stress. So in these studies, We are doing PK analysis of the ampicillin through the core that Jeremiah and Brookie lead, milk oligosaccharide analysis, mother's milk oligosaccharide, we call it, since we have mice here, not just humans. And that is through the BOTA core. We are analyzing the composition of the infant microbiome and metabolome, working with our core in our division as well as Peter's lab. And then we are going to assess the functional impact of that, studying whether subsequent challenge with group B streptococcus. Interestingly, the rate of late onset group B streptococcal disease has not changed at all. Some data suggest it's slightly increased in patients who've had GBS prophylaxis. So we'll challenge them with group B strep and evaluate their innate immune cell function, and then also evaluate, and this is the expertise of the Liu lab in particular, who studies vaccine development for high priority pathogens and the associated adaptive immune response. We're going to be looking at the hepatitis B vaccine and the pneumococcal vaccine and see what type of immune responses, whether there's T cell skewing towards Th17, Th1, which can dampen immune responses, versus the Th2 Treg phenotype, which is seen to be more favorable towards antibody responses. Next slide. And I think that's where we end. I'll just say that we've run through two different sets of mice. We have two very talented project scientists, Chi-Ming Tsai and Fatima Askarian, who are working on that, and they'll be able to present their research. They've learned how to milk mice from the BOTA lab, which is not as easy as you might imagine. We're finding that the repeated antibiotic administration to the mothers has led to some stress where they become a little less effective in their nurturing of the infants. That we're trying to optimize and maybe relax our dosing regimens a little bit. And I see a question in the chat. Have you done studies where you reintroduce the gut flora and check whether it reverses the metabolome changes? We ourselves have not initiated that, but there is some excellent work going on from Eric Paymer and other groups where there is the evidence that autologous restoration, basically banking your own flora, knowing that you might be going into a window of high risk of antibiotic exposure, and reinstituting it can be beneficial in settings like bone marrow transplant. There's also evidence that basically swathing the baby in like a C-section delivered baby with some vaginal secretions that they may otherwise have could transmit some of the flora that may be necessary for restoring stuff. Mark Murphy, have you considered investigating oral beta lactamase? That is very interesting. There's actually some outstanding research on that. If this was investigated as a potential C. diff preventative, and I think the idea is that you could treat systemically, achieve your therapeutic benefit, but then administer an oral lactamase to protect against the dysbiosis. I can see how that might work in the mother, and but you're also going to get systemic exposure in the baby, and it may be hard to administer that just at the outset. But that is a very, very clever approach that I've seen published recently. If you used, we are just using from Varsha the question about combination therapy, AMP and GENT, or AMP and cephalosporin is the common prophylactic antibiotic regimen. We're, for unanimity with the other two treatments, we are just looking at the ampicillin there. We believe that the cephalosporins, of course, added to provide excellent CNS coverage in these high-risk infants, and the aminoglycoside has synergy against streptococci and some other organisms. We would expect that the impact on the microflora would be greater, and it might be hard to distinguish whether it was one or the other drug, depending on the susceptibility. Ampicillin, probably of all those things, has the greatest anaerobic coverage, and so much of the gut flora is anaerobic. Is it possible to check whether these metabolome changes are not the direct effect of the drug itself? So this would be an independent effect, not on the gut flora. That could be studied, I guess, to see if we had any added effect in enotobiotic mice. And we're certainly looking to see whether the effects of the antibiotic on the liver metabolism might subsequently lead to other metabolome changes, actually running some experiments in uninfected mice to provide to Shirley and Peter for those analyses.
Any other questions for Victor?
Okay.
All right, so if we could get the slide deck back up, I think I'll hand it over to Peter Dorstein and Shirley Sonoda that head up our data science project.
Great, thank you, Audrey. As Audrey mentioned in the beginning, our data science project sort of interfaces with both the clinical project that Tina's leading, as well as the basic science project that Victor and George are leading. We are, next slide, please. I'm just going to outline our three aims. So our first aim, we have some data, previous data, both in the Dornstein lab and then together, Peter and I collaboratively, where we can monitor basically the chemical metabolome of humans by using non-invasive skin swabs. So we felt that this was a particularly good population to utilize these non-invasive skin swabs. They're literally just soaked in an alcohol solution and non-invasively rubbed on the skin, various different places. So we're going to be using these skin swabs in our clinical study to monitor drugs, particularly antibiotics, and we are also able to see metabolites of drugs on the surface of the skin, both in the mom and the baby. Next slide. And then we will be determining the impact of this antibiotic exposure on the infant microbiome, the metabolome, and also on metabolic transformation capacity. Next slide. And then finally, we will be looking at our gut metabolic transformations more directly in the animal model by mimicking the clinical study as Victor outlined, by looking at ampicillin administration transplacentally through breast milk and then directly to an infant mouse. And we will be able to look at both intestinal and hepatic cytochrome P450 activity in that mouse model. Next slide. So our overall hypothesis driving these studies is that we believe that indirect administration of antibiotics could impact the developing infant. So breast milk exposure that we are going to be monitoring non-invasively in the skin, we believe does have a direct impact and a significant impact on the infant microbiome and the infant metabolome. So we'll be demonstrating the feasibility of these non-invasive skin swabs to monitor this. And we will also be measuring the impact of these antibiotics on both the microbiome and the metabolome of the infant. And hopefully we will be able to understand how the infant metabolism is altered with maternally administered antibiotics. Next slide. So thank you. So as Tina outlined in our clinical study, we are currently collecting all of these clinical samples. Next slide. And while we're waiting, we have done quite a bit of additional work to sort of support these studies. Next slide. So we have a large database, a publicly available database called GNPS, Global Natural Product Social Molecular Networking. And we have been able to tap into this database to look at our human milk, our existing breast milk samples in this database. And this is work done by Sydney Thomas, and this work is currently being under review for publication. So she took almost 1,000 human milk samples and ran it through our untargeted metabolomics pipeline. Next slide. And what you see here is an example of what our untargeted metabolomics pipeline can do. We have lots of analytical capabilities within the database of GNPS, and we are able to not only identify some drugs, lots of drugs actually, but we can also apply a technique called molecular networking, where we look at closely aligned mass spec spectra and try to determine whether or not some of the related molecules may be metabolites of drugs. So in that left-hand panel, Sydney was able to determine that there were a fair number of sulfonamide antibiotics and their metabolites seen in the breast milk. And in that lower panel, she saw drugs, or she saw amitriptyline as well as the active metabolite nortriptyline in breast milk samples. So the majority of drugs seen in the breast milk samples were either antibiotics or antidepressants. And then interestingly, in that lower portion in the middle there, she saw some bacterial quorum sensing signals that in the breast milk as well. Next slide. And then she also took a sample of, again, in our publicly available database of GNPS, she found 42 paired mother-infant dyads who had not only breast milk samples, but infant oral samples, infant skin samples, and infant stool samples. And focusing on antibiotics, she found interesting, she's found some interesting data with azithromycin and clindamycin as well. So she found matches in our GNPS database with these antibiotics. And if you can kind of focus your attention on the right-hand side, that graph there, interestingly, she saw some paired infant data with the mother's breast milk and the infant's stool. So the light colored tan circles and the light blue circles show samples that were paired that showed both antibiotics in the mother's breast milk as well as the infant stool. And interestingly, she saw an azithromycin metabolite both in the breast milk and the infant stool, suggesting that that antibiotic traverses the breast milk into the infant and is seen in the infant stool. Next slide. So I'm not going to steal Sydney's thunder here, but we received a support pool grant, and we're currently working on another set of human breast milk data where we're applying a multi-omic analytic technique. And she's going to describe that tomorrow in the support pool grant mechanism. Next slide. So while we're waiting again for the clinical samples, we're working on the animal AIM, which is AIM 3, together in collaboration with the Nizay and Liu lab. Next slide. So as Victor mentioned, we're giving ampicillin to mouse mothers, both to mimic placental and breastfeeding administration, as well as direct ampicillin to the infant mouse. And we've completed the placental transmission, and I'm going to show you just some very preliminary data from that. Next slide. And our questions that we're trying to answer in this AIM is, how does this affect the infant intestinal and hepatic cytochrome P450 gene expression, protein levels, and enzymatic activity? So we, Victor already mentioned Alison Brabenek's data, and then we have data in adult humans where we showed that giving an antibiotic may decrease cytochrome P450 enzyme activity that we showed through the administration of the Cooperstown cocktail that was, proxy for cytochrome P453A, 1A2, 2C19, and 2D6. Next slide. So we're hoping to investigate this more directly in the mouse model. And we're going to utilize our untargeted metabolomics and molecular networking data analytic structure in combination with targeted analysis of ampicillin in our PAC core run by Jeremiah and Brookie. And then we will also incorporate microbiome sequencing utilizing our microbiome core here at UCSD run by Jack Gilbert. And then we will be processing liver intestines to, again, look at regulation of cytochrome P450 activity. Next slide. And we will also be applying neural network strategies because, as you can imagine, this is a lot of data. And trying to make sense of what's going on with all of this data is quite a job. And Peter Dorenstein and Rob Knight have pioneered some amazing neural network strategies to try to make some sense of the relationships between what's going on with the microbes and what's going on with the metabolome. Next slide. And next slide. And Fatima in the Nizay lab has been working very hard on these breeding the mice, and she's given us samples already that Sydney and our team have done some preliminary analysis on. Next slide. So this is just showing you the mother's samples, mother's fecal samples of ampicillin treated animals versus control. And again, just to kind of give you a high order view that there are signals going on with the ampicillin treated mice that are not seen in the control. And some of those graphs that you see don't have annotations quite yet, but we do see ampicillin, which is a good thing. So in the dark blue circles are annotated for ampicillin, and the light blue circles are thought to be molecules, metabolites that are related to ampicillin. So we're still working on identifying all of those. Next slide. And then as you take, this is these are PCA plots, and going from left to right on the top and then left to right on the bottom in time, you can see that day -3 before the ampicillin is given, the control and the ampicillin overarching view of the metabolome looks similar. And if you point your eyes to the day 21 after antibiotics should be gone out of the system, you can see that the PCA plots also look similar. However, as we march through in time, it looks like the ampicillin treated animals are diverging with respect to the metabolome. And so Sydney has identified, for example, bile acid molecules, as well as some other metabolome changes that are driving possibly these differences in the ampicillin treated versus the control treated animals. Next slide. And finally, just to sort of drive home the point, this is infant fecal data that, you know, even if we don't necessarily see a lot of ampicillin in the feces, that the antibiotic may be driving changes that we see in the infant metabolome. And so again, just marching through day 7, 14, and 21, you can see that in the green colored bars, the ampicillin treated mice are quite divergent from the control mice. And for example, if you see that right-handed graph on the top, they differ in terms of purine nucleosides. And then down on the bottom, the phenyl ether metabolite may be different in the ampicillin treated mice versus control. Again, just suggesting that we may see changes in our infants due to antibiotics that are not necessarily some things that we can predict or that we would expect. Next slide. So what are our next steps? We're going to continue this multi-omic analysis of our breast milk samples and integrate infant developmental outcomes into this analysis. We're going to be continuing our mouse studies investigating the antibiotic and microbial impact on drug metabolizing enzymes. We're going to be hopefully analyzing 2,500 breast milk samples that from Tina's human milk biorepository. And this is hopefully funding that we're going to be receiving through the administrative supplement. And then finally, we are continuing, the Dorenstein lab is continuing to create more data science tools that we will apply to the maternal pediatric community. And now I want to just turn it over to Peter to excite you with some of those developments.
Great, thank you. It's been a wonderful morning so far, and Shirley already highlighted molecular networking a little bit. And in our case, one of the fundamental questions that we often have, and I think that was also one of the questions during Victor Nizet's talk, was how do you figure out who is causing the change in the metabolome? And I think that's a fundamental question. So if you treat with antibiotics, you change the microbial community. Is it really because the metabolome is changing because the microbes are digesting different molecules or metabolizing different molecules? Or is it maybe a change that is caused in the host because of the changing microbial community and exposure to different molecules that the microbiome makes? And so in this case, what I'm showing you is how we actually discovered new bile acids that we published on in 2020. Also, Victor Nizet highlighted it in his example. But these are, instead of glycine conjugated bile acids, we see phenylalanine, tyrosine, and other amino acids that are conjugated to different free acids or free bile acids. And in our case, we observed them to be produced in mice that are colonized with the normal microbial community, and they would be absent in germ-free mice. And so the fundamental question that people often will ask is, well, you're working in an animal study. Is that actually translational to humans? And that's a problem that we are really interested in. And so we're developing a number of different tools, and I'm just going to highlight 2 of those tools. So next slide, please. And the first tool is we call MAST. It's a mass spectrometry search tool. And basically what it allows you to do is take these mass spec signatures, whether they're known or unknown signatures, and you can find what are the data sets it's found in. Next slide, please. And we'll have an animation, just hit it 8 times. But basically what we've built is a knowledge base And the way you have to think about this is that we build a knowledge base and a search engine. And the way to think about it is that it's similar to a Google search where you can put in text. In our case, instead of text, we put in mass spec data features. And then we can search to what data it's matching to because we have this knowledge base. And so we can learn about NCBI taxonomy, age, disease ontology, Uberon ontology, which is organ system, biofluid, sex, longitude, altitude, depth, if that information is available, and mass spectrometry ontology. And so next slide. Next slide. Maybe. Is it stuck? All right, I'll just keep going. So when we search with these particular bile acids, what's really unique is that we find lots of human data sets where these new bile acids are found. And then when we map it in relationship to disease ontologies, we see them quite abundantly or frequently associated with IBD, Crohn's disease, ulcerative colitis, as well as other metabolic diseases such as diabetes. And so that once you're able to find this, so this is the observation associated with diseases. Once you have these observations, now you can go to those public data sets and say, okay, let's look at the metadata associated, deeper inspection of the metadata associated with those diseases. So let's go to the next slide, please.
بس ما زي يتفكر هذا.
All right. So anyway, so we look deeper into a pediatric IBD cohort for the levels of these different bile acids that are existing. And in this case, it was plus or minus antibiotics that was associated, had the metadata of this with it. And what you see on the left, you see the taurine conjugated bile acids. Those are well-known human derived bile acids. And they actually get converted into these new bile acids. It's not published yet, but we have the mechanism by which it's done. But on the right, you see the histidine, phenylalanine conjugated bile acids. And upon antibiotic treatment, you see the decrease of these particular bile acids. And so, but the question still remains, is it the microbes that make them, or is it due to an induction of enzymes of the host or humans? And so we're developing a new tool. Next slide, please. We're developing a new tool called MicrobeMass. And basically what we've done is we've taken 58,000 cultured microbial data and then mapped that onto the NCBI taxonomy. So now when you search with that mass spec feature, next slide. And this is my last slide, and it will map it onto the phylogeny associated with all these mass spec features that you're searching with. And so in this case, it's the phenylalanine conjugated bile acid. And what you then can see is how it's phylogenetic distributed. And you can see that it really is made by microbes because these are in culture. And if you hit one more forward, you'll see that these bile acids are made by bifidobacteria. Now, these are early colonizers of infants. They particularly come up during breastfeeding. And one of the things that we've learned so far is that they control IL-17, they control Tregs, they control IL-10. So I think there's going to be a story that might emerge is how these are regulating the infant immune system, but that is also manifested through antibiotic exposures. And so I just wanted to let the community know that these tools are available. I'm happy to work with everybody, and we look forward to working with this community.
Thank you, Peter. Shul, I think there was a question during your talk that came up in the chat from Kate Stika. She said, when you were looking at changes in the CYP activity with the cocktail and the AMP exposed folks, are you also controlling for changes in inflammation, which may occur due to labor itself?
Yeah, that's a great question. And, you know, we know that inflammation can affect the cytochrome P450 activity as well. I would say in the mice, we're doing as much as we can to control their environment and treat the control and the AMP exposed mice similarly. But what I will say is that using our untargeted metabolomics analytics, we can probably look for some of the markers of inflammation because we can see so many endogenous chemicals in our untargeted metabolomics analytics. So, it would be definitely something to look for that, could be a confounder.
Great, thank you. If we could have the slides again, please. So I'm going to go ahead and on Lars Bodie's behalf, go through the milk analytics core. In these last few minutes, we'll do our 2 cores that we've got here. So really the cores for Imprint and our center are just like what was presented before by the Hub and the KRCC, as well as the Vanderbilt group. We're really here to support the work that's going on in the clinical basic science and the metabolomics studies that are ongoing. So the first aim that the Bodie Lab is working on is providing human milk specific collection protocols and kits for the composition analysis, and then really developing and validating these specific assays to expand the analytical capabilities of the program. Next slide. So you can go ahead and continue the animation on this side, please. So human milk itself is actually, we don't have to preach to everybody here, but is really a dynamic biological system in and of itself. So there's, just as Aaron Pollock pointed out from the very beginning, there are knowledge deficits in regards to the study of human milk, and so that creates really important research opportunities. And you can see that the goal here is as kind of that main figure that we have as kind of the center of the work that we're doing here with the mother infant human milk. You can see that our ultimate goal, of course, is to understand human milk better and therefore improving maternal child health. If you can go on to the next slide, please. So it's not trivial to study human milk. I mean, Lars and his lab have made a career out of it. It's dynamic, it's a complex matrix. And so there's unique molecules. The Bodie Lab really focuses on human milk oligosaccharides, which represents the third most abundant component of human milk after lactose and lipids, and really understanding what these HMOs are capable of doing and how they are something that's unique to human milk that are difficult to produce and just to really understand. So that's why there's really a focus here on protocol development and assay development. Next slide. Again, as I mentioned at the outset, the aims here really focus on supporting the clinical and data project that you've seen there, that you've heard of already, as well as the oligosaccharide and microbiome analysis for the research projects. Next slide. And then in aim 2 with the analytical capabilities, it's just trying to have more multiplex immunoassays in human milk analysis, and then really serving as a resource, which I hope that those of you that are interested in this area that Lars is working in, we can certainly put you directly in contact with him for the analytical assays that he's developed for human milk. Next slide. So here the vision is to become a comprehensive milk analytic core with the ability to do this sort of analysis. There is the MOMMY project, which just came up very briefly on the previous slide. I apologize for racing through that, but Lars really oversees a huge biomilk repository there, where there's a mother milk infant center of research excellence that is supported by the Larson Rosenquist Foundation. So next slide. And with that, in the few minutes that we have left for our center, I'll hand things over to Jeremiah Mompers to talk about our pharmacometrics and analytical chemistry core.
All right, thanks, Audrey. I'm thrilled to see so much participation in the Imprint inaugural meeting and glad to have a few minutes to talk about our core. I direct this core along with Brookie Best, and we also work closely with our colleague Edmund Caparelli here at UC San Diego. We're one of the 6 Imprint Hub scientific cores, and we provide bioanalytical support and PK/PD analyses for our local UCSD projects. And we're also available to assist the broader Imprint network, as well as applicants for opportunity and support pool funding. Just to provide a little practical context on our laboratory, our footprint consists of 5 triple quad LCMS systems, a GCMS for working with volatile compounds. We have an ICP-MS for measuring heavy metals, a MALDI-TOF biotyper for microbial confirmation, and then a ThermoQ Exactive Plus high-res mass spec for identification of unknowns. So as a general distinction from what Shirley and Peter described, their work in molecular networking and metabolomics is primarily untargeted, whereas in our group, we focus on targeted analyses. In other words, quantitatively measuring drugs, metabolites, and biomarkers in different biological fluids and tissues. Next slide, please. So the infrastructure enabling our core really lies within the UCSD Pediatric Pharmacology Laboratory, or PPL. We are a pharmacology support laboratory within the IMPACT network. We've heard a little about IMPACT already today, and Sharon Nachman, the IMPACT network chair, is going to be speaking later this afternoon. Within IMPACT, our lab has been performing quantitative assays of antiretroviral drugs for over 15 years. The lab is CLIA approved for high complexity testing. We participate in proficiency testing programs, et cetera. And we can operate as a GLP facility when required for studies that involve regulatory submissions, for example. Dr. Pollack this morning in his introductory remarks mentioned the role that Imprint can play in training the next generation of scientists. And we think we're well positioned to contribute. We do provide an ideal training environment for students and postdocs associated with Imprint who wish to gain exposure to quantitative bioanalysis or PK/PD. We also are involved in several existing training programs that can be tapped into for additional synergy. Those include an NICHD supported short-term research training grant, T35, that Dr. Best directs. We also have an industry-supported 2-year postdoctoral fellowship in pharmacometrics that I direct. That's with Pfizer. And we also have a critical path grant with the FDA's Division of Pediatric and Maternal Health. That's directed by Tamara Johnson. And I'll speak a little bit more about that in just a minute. Next slide, please. So just taking a short while to talk about some of our current activities and broader research interests. First, our core is working on developing a standardized analytical platform to measure drugs in milk. The rationale for this project is that there can be tremendous variation in milk composition, as opposed to, for example, measuring drugs in blood or plasma, where the matrix tends to be relatively consistent from sample to sample. Often when drug concentrations in milk are reported, it's more of a one-size-fits-all approach that doesn't take into consideration variability in breast milk composition, such as differences in lipid and protein content, milk phase, and stage of milk production. We hope that this platform may better describe the scope of drug and metabolite concentrations in milk for a particular drug, and therefore be better suited to estimate infant exposures and inform trial design and sample handling requirements for future clinical lactation studies. Just as a quick example, we recently measured tetrahydrocannabinol, or THC, the main psychoactive component in cannabis, in breast milk samples from self-reported cannabis users. As shown in the figure, there's significant variation in THC levels, with some levels above 1 microgram per mil. And when we defatted the milk samples, we saw that THC was essentially absent. This is, of course, what would be expected for a lipophilic compound such as THC, but it does highlight some of the associated variability, which can be due to lipid content of individual milk samples. Next slide, please. Our team also has a longstanding interest in the application of PBPK in pregnancy. As we heard this morning, there's interest really across Imprint in PBPK, including in the pharmacometrics and clinical trial design core with Sarah Quinney and Rob Bees, as well as through a collaboration with the Bill and Melinda Gates Foundation. At a very high level, PBPK is a versatile computational tool that combines drug characteristics and physiology to develop a mechanistic understanding of to predict drug exposure and effects. And I think there's good reason for such enthusiasm around PBPK in pregnancy, in part because it integrates physiological parameters that tend to change quite a bit with time across pregnancy, things like maternal body weight, organ volumes and blood flows, etc. As I mentioned before, we're also part of a critical path grant with the FDA's Division of Pediatric and Maternal Health. And this grant primarily focuses on PBPK in pregnancy. This program has funded pre and postdoctoral fellows working in this area. The photo on the bottom shows our past fellow, Mary Gockenbach, presenting her work at FDA's Science Day. And shown on the right is a schematic of a PBPK model structure, just highlighting the complexity of these models. This arrangement also includes additional pregnancy relevant compartments, including the placenta, the fetus, and arterial and venous cord blood. And this was led by Mei Liu and Andre Dahlman, some of our collaborators in this space. Next slide, please. Our group has also had a recent focus on looking at so-called washout concentrations of transplacentally acquired drugs in neonates. Largely, this has been with antiretroviral drugs. For example, within the IMPACT P1026S study, we had available opportunistic samples taken in newborns after birth, which can be used to estimate elimination in the baby. The data shown here are for the integrase inhibitor elvitegravir, showing samples in cord blood and opportunistic samples between 2 to 10 hours, 18 to 28 hours, and so forth after birth. So these data may be used to better understand the activity of particular enzymatic pathways after birth, for example, CYP3A activity in newborns, and can also be used to simulate different dosing regimens during the first days of life to design clinical trials. Next slide, please. So this slide illustrates that approach. In particular, this is related to the drug dolutegravir, which is an integrase inhibitor widely used for the treatment of HIV. Because children born to mothers with HIV typically should begin ARV regimens as soon as possible after birth, the dosing of dolutegravir in newborns is important, although the PK of this drug has not been yet been evaluated in this population. So we used maternal PK data and washout infant data to develop separate maternal and neonatal population PK models, which were linked by the neonatal concentration at birth, which in turn was a function of the maternal concentration at delivery and the cord blood to maternal plasma ratio, which we estimated with available data. Because dolutegravir is primarily glucuronidated by UGT1A1 and the activity of this enzyme is very low at birth, we showed that newborn dosing requirements during the first days of life may in fact depend upon maternal dosing history prior to delivery. So in other words, if the mother took the dose 6 hours prior to delivery versus 24 hours prior to delivery, this difference could impact when dosing should be started in the infant. And overall, these simulations were useful in designing potential strategies and partially formed the basis for a clinical trial, which will begin enrolling soon. Next slide, please. And I'll conclude just by highlighting a few of our other additional maternal pediatric research areas within our core. First, we have an interest in the postpartum period, in particular focusing on the question of when PK parameters return to quote unquote normal in the mother. There's a general assumption that postpartum is essentially equivalent to non-pregnant from a pharmacokinetic point of view, and certainly that's not entirely true with the significant physiological changes that happen postpartum. For example, the figure on the bottom left shows the trajectory of albumin in 354 postpartum women. And you see a sharp increase after delivery, but that's not instantaneous. It takes several weeks. This work was a collaboration with Anil Maharaj from the University of British Columbia and our fellow Sherry Zhao. We also have an interest in protein binding and better understanding free drug concentrations during pregnancy, recognizing that unbound drug is responsible for therapeutic effect. Historically, pregnancy PK research has primarily focused on total concentrations, but those don't tell the whole story given how albumin and AAG concentrations can change during pregnancy and alter protein binding. We have an active R21 in this area measuring unbound concentrations from a number of completed pregnancy PK studies. And that work is a collaboration with Tim Cressy from Chiang Mai University in Thailand. Finally, our group is also involved in a number of COVID-related research projects in pregnancy. Pregnant women were largely left out of drug studies for COVID early in the pandemic. And as a research community, we're playing catch up to some degree. Within Impact 2032, we're working to understand the PK of remdesivir in pregnant women, remdesivir being the first antiviral drug that was FDA approved for COVID. Showing here the preliminary results presented by our collaborator, Christina Brooks from the University of Colorado at Croy this year. And finally, we're also working with Gilead and the UCSD Human Milk Biorepository to characterize the breast milk transfer of remdesivir as well as its metabolites. So I'll conclude here. Thanks so much for your time, and please reach out to me during the meeting if you'd like to chat about any of the capabilities of our core. And I think we can take questions at this point about any of the UCSD presentation.
Thank you, Jeremiah. There is one quick question. Any difficulties to achieve a stable baseline LCMS for the cannabinoids in the respective matrix?
I'm not certain what the question relates to as baseline. Could you expand on your question, please? Otherwise, we can revisit that. I'm not certain exactly what's being asked.
And there's one other question. In the research work using the GNPS, how are you able to distinguish changes in infant stool sample substances that are related to acute viral or diarrheal illness from those with ampicillin exposure?
So it's a great question. So the way to think about GNPS is that it's a data repository and an analysis ecosystem. So what GNPS together with the mass that I highlighted, it allows you to find specific molecules that might be of interest. And so if there are acute viral infections or diarrheal illness is associated with data sets that are in the public domain, now you can go deeper into those data sets. So I know that there's some viral infections such as Zika virus that have been investigated. There's a couple of other ones that have been investigated. And in the case of some of the bile acids, we do see some of the different bile acids that are upregulated or downregulated depending on the viral infection. Diarrheal disease, the only one that I'm familiar with is a human. I mean, it's not really a diarrheal disease, but it's somewhat related. It's a pediatric where they're pancreatic insufficient. There's a data set of that. And in those cases, we do see an increase in terms of some of the bile acids that we see as well that are made by these microbes. But there's no relationship of those studies to ampicillin exposure.
Thank you, Peter. So in the interest of time, I think we'll hand things back over. I think Lang, your group is up next. If there's other questions, please direct them to members of our team. We'll be happy to answer in the chat. Thank you, everybody.